Hey biology students, in this video we're going to cover antibodies, which is a part of the third line of defense. Recall what cells secrete antibodies. That's right, plasma cells. And plasma cells are a type of B lymphocyte. Let's watch this quick animation to review the process of the humoral response when antibodies are secreted.
In the other adaptive immune response, humoral immunity, the immune system targets pathogens circulating in extracellular fluids, including blood and lymph. Invading pathogens, such as bacteria, are detected by specific white blood cells called B cells, which recognize specific antigens on bacterial surfaces. Once B cells are activated, they proliferate and differentiate into plasma cells, which secrete millions of antibodies that circulate throughout the body and activate several defense mechanisms. In one method, antibodies that bind antigens on the surface of a pathogen can inactivate or neutralize it by interfering with its ability to infect a host cell. They can also opsonize or tag pathogens for engulfment and destruction by phagocytes, such as macrophages or neutrophils. Finally, antibodies can activate the complement system, a complex of proteins that further enhances the opsonization and destruction of pathogens. Even with the pathogens destroyed, some activated B cells differentiate into memory B cells instead of plasma cells. These memory B cells continue to produce small amounts of antibody long after an infection has cleared. If the same pathogen re-enters the body, these circulating antibodies can target it for immediate destruction.
Okay. So these antibodies are actually proteins secreted by the plasma cells. Let's take a closer look at their structure. So antibodies are composed of 4 different protein chains. There's 2 heavy chains. So the heavy chains are shown in purple. So here's one heavy chain, and here's the second heavy chain. These heavy chains are held together by disulfide bonds. So there are actually disulfide bonds that hold the chains together. Then there are 2 light chains. So the light chains are shown in green. So there's a light chain and there's a light chain. So composed of 4 polypeptide chains. Two heavy. They're called heavy because they're longer. So you see the purple ones are composed of 4 subunits, and the light chains, the ones in green, are composed of 2 smaller subunits, so they're smaller. So 2 heavy, what we call 2 heavy and 2 light. And the important parts of this antibody protein that you should be aware of, one is that there's a region called the Fc region, which is basically the stem of the antibody. So when we draw these, of course, we usually draw them as just a capital Y. So the stem would be the Fc stem or Fc region. And basically that just means the C is for constant. So it's a constant region that pretty much is the same from species to species. So all animals produce antibodies in infections. And so in humans, that Fc stem is a conserved region and among other humans. And then, if we look at an antibody secreted by a mouse or a rat or a horse or a dog, well, that Fc stem is going to be variable from species to species. Now, the region that's variable from antibody to antibody is what's represented here by the tips of the letter Y. And here you can see the antigen binding sites at the ends there. Let's take a highlighter and let's highlight what we mean by the antigen binding sites. So let me move these markings. So these are the antigen binding sites. So we're going to highlight those in yellow. So those are the antigen binding sites. So those are at the tips of the Y. It's at the tip of that heavy chain, but it's also at the tip of that light chain as well. So sometimes some people actually draw it like this when they draw the antibody and show that this whole region is an antigen binding site, and this whole region is an antigen binding site, and that's where the specific antigen is going to bind in the antibody interaction. So this is what the protein model looks like over here on the screen. Let's give an example of how antibodies interact with specific antigens. So I took these two pictures of the influenza virus, and let's say that you were vaccinated against influenza. You got the flu vaccine in, let's say, 2018. Flu vaccine. We will cover vaccines in the next video in more detail. So let's say you had the 2018 flu vaccine, and that flu vaccine had this particular strain of influenza in it. Okay, and there's a reason why we have a flu vaccine every fall that's available. So it's always recommended to get your flu vaccine right around October of every year. So I say, remember when it's Halloween, you should be thinking, have I gotten my flu vaccine or not? Because really, that's the scary thing. Halloween is a scary holiday, but what's even scarier than Halloween is not being protected fully from influenza virus. So in 2018, let's say you got the flu vaccine and it protected you against this particular strain of influenza, which we see pictured as a yellow flu virus, or sorry, not yellow, green flu virus. Okay, so that means that your B cells would have produced antibody against, we'll say we make a whole bunch of these, and I got to do the stem region first, and then I'll come in and I'll make the antigen binding site portion. And really, we need to show that those antigen binding sites do actually fully attach to the antigen. Okay, and then we'll have some more up here. And so what I'm trying to show you, the antigen binding sites in green. So the antigen binding sites are what binds specifically to the antigen, and the antigen would be some protein component of this influenza virus here. So you get vaccinated in 2018, it protects you against this strain of the influenza virus. Usually, by the way, it actually is a collection of a few different viruses where you're gaining immunity to maybe 2 or 3 of the most common influenza strains that are circulating the globe for that particular year. But to make it simple, we'll just say it's this one, it's this particular influenza. And it's the same thing as if you had actually gotten sick in 2018 with the flu. Well, you also would have developed memory cells, just like vaccination. You just would have had to go through the whole awful experience of being sick, but you also would have memory B cells that also would have produced antibody, And this antibody recognizes the particular antigen of this strain of this 2018 influenza virus. So then it's 2019, and that was the most recent flu vaccine. So now let's say it was 2019 and you got the flu shot again. And why do we get it again? Well, let's take a look at the flu in 2019. Purple instead of green. What is that really representing? Really, that's showing you something that many pathogens are capable of doing, which is they can change their antigens. And they do this through a process we call mutation, which, whoops, I know you have studied. We've studied together. So that means that there's mutation in their genetic code. So there's mutation in the genetic code of the virus, and then that leads to antigenic variation. Basically, we end up with new antigens on the surface of the virus, different enough that these antibodies over here that recognize and bind to the 2018 influenza, they are not going to be able to bind to the 2019 influenza virus because they simply do not have the same component. They don't have the same antigen anymore on the flu virus. So only that original green antigen could be identified by the green antibody. So that means that either you need to get a new flu vaccine, and that's why it's recommended to get the flu vaccine every year in the fall, right around Halloween, to protect yourself from that particular year's new and improved version of the influenza virus. So recommended you do that. And then what happens is, well, then you make a different type of antibody. This time the antibody would be specific to the purple antigen here. So the antigen binding site. So we would get an attachment there. And here's another one being made. And so we would get the antibody, the specific antibody recognizing this new antigen. And that is actually a feature of a lot of pathogens. And it's sort of like if you watch any of those crime scene investigation shows where there's a particular criminal and the police put out an APB, find this particular criminal. He is 5'10", Caucasian, wears glasses, right? There's a pretty detailed description about who the criminal is, and everybody, all the police are looking for that person. That's like the green influenza. So all the police, being these antibodies, are looking for this particular version of the influenza virus. It looks this way, it has this particular shape and the protein component. So we're looking for this person, that has this particular appearance. Well, what do these criminals do sometimes in the show? Well, yeah, you see them go into a bathroom and they've bought some hair dye and they go ahead and color their hair a different color. They might even cut their hair. They might do other dramatic ways to change their physical appearance because then they don't match the APB anymore. Now everybody's looking for somebody who's blonde hair, who's 5'10", wears glasses. That was the APB. That's what everybody's looking for. But hey, this person has actually now has black hair and a nose ring and looks really different. Lost the glasses, not wearing glasses. So that's what we mean. The pathogen can change its appearance to the immune system, and that always puts the immune system a step behind. So now let's talk about actually some different classes of antibodies. So we've been using this word antibody Recall, though, that you can use another word. The other word you can use is immunoglobulin. So if you see the word immunoglobulin or antibody, they are synonymous. They mean the same thing. A lot of times we show immunoglobulin as capital I, lowercase g. So there are 5 classes of antibodies that you should be aware of here. We're just going to look at the slide for this, which you should have in your lecture module. So IgG is the most common antibody, and it is very small. It's actually a monomer. So when we draw the antibody here, and remember I oftentimes just use a capital letter Y. It's a simplified way to draw the antibody. This picture is a little bit better because it shows the 4 polypeptide chains connected. So when we draw it as a simple Y, capital Y only, then we refer to its structure as monomer, meaning it's only one antibody structure. Because it's so small, this is the antibody that crosses the placenta, and it offers the protection of the fetus during the development stages. IgM is actually the largest antibody and the first antibody made in response to antigen. It has a pentamer structure, and that word pentamer, pent means 5, so that means you actually have 5 of the Y, each capital Y, you have 5 of them in this pentamer structure. So here's a question. If we have 5 antibodies that make up one molecule of IgM, how many antigen binding sites does IgM have? That's right, it has 10. So there's 10 antigen binding sites in the IgM molecule. It's huge. It's a huge molecule. It explains why it's actually the first one that's made in response to antigen, because it's your largest tackle football player, is how I think of IgM. IgM is the tackle football player. He's going after a pathogen, and boy, he's going to grab onto 10 of those guys at once. So that's a big advantage of producing IgM first. IgA, this one hangs out in mucous membranes, so it's in body secretions. Mucus, saliva, tears, breast milk. It's a dimer. So dimer means there's 2 antibodies molecules connected to make the IgA structure, and that means there would be how many antigen binding sites? 1, 2, 3, 4. 4. How about IgE? IgE is not present very much. It is actually involved in allergies. So people who produce more IgE antibody than others may be more prone to allergies. And what IgE can do is actually bind to those cells that release histamine and can initiate an inflammatory response, basophils and mast cells. And it's a monomer. Okay, IgD, this is our last class of antibody, monomer structure. This is what's attached to the surface of the B cell. So this is actually the B cell receptor, the BCR. So when we draw the B cell, which we did in the last video, and we showed the receptors on the surface of the B cell, well, that was IgD. All right, next we're going to talk about the functions of antibodies. How do antibodies assist in eliminating the pathogen? So first thing to recognize about antibodies is that this is a different type of attack than the cytotoxic T cells. And remember, it is in a separate branch of adaptive immunity. And this branch of adaptive immunity attacks extracellular pathogen. So what do I mean by extracellular pathogen? I mean that these are pathogens that are outside of the cell. That's what extracellular means. It means that they're outside the cells of the body. So they're outside the cells of the body. So your body cells, they are not in the cell. They're outside of it. And they may be floating in blood or in lymph or in tissues, but they are extracellular. And so the antibodies are specifically designed to go after, like little magnets, grab onto extracellular pathogen. And that's different from what the cytotoxic T cells do. Cytotoxic T cells bind to infected body cells, where those infected body cells are harboring intracellular pathogens, pathogens that are inside of the cell. Make sure you understand that clear difference. Okay, so if the antibodies can bind to extracellular pathogen, or extracellular antigen, really is what it is, then what do? Well, the first thing that they do is neutralize the pathogen. So to neutralize it means to prevent it from attaching to any target cells. So this could be in the case of virus. Virus is considered an intracellular pathogen. So it's considered an intracellular pathogen, meaning that in the previous video, we talked about how viruses go inside of cells where they reproduce inside of the cell. That's what intra means they're inside of, this would be inside of our cells, human cells. And they do, but also they spend some time outside of the cells. Once they lyse out of the cell, then they are floating in the body fluids. And that would be the prime time for virus to grab it. I'm sorry, for antibody to grab it. So if antibody grabs it, then we say it's neutralized. So we can neutralize a virus, we can neutralize a toxin. A lot of bacteria produce toxins. And so those toxins have to bind to target cells to have some sort of physiological effect. Well, the antibody is going to prevent that from happening, neutralize it. And then over here in the third picture, we're looking at a bacterial pathogen where we have a bunch of antibody bound to the flagella, and that's going to prevent the bacteria from swimming efficiently. So neutralize, neutralize, neutralize. All right, function number 2 of antibodies. Here we're looking at IgM, that big giant tackle football player antibody, the pentamer, and it's showing us it can agglutinate. So it's agglutinating, and it's binding to more than one bacteria at a time, and even sometimes more than one epitope at a time. And that just, agglutinate just means to clump. So it's making a big clump of bacteria, grouping them together so they can't migrate through the body. Third function, opsonization. And this has to do with remembering that the stem of antibodies is called the Fc stem. So when we draw an antibody and we talk about the stem, this is the Fc stem, constant region. That also happens to be a binding spot for phagocytes. So they're showing that there's an Fc receptor on the surface of macrophages as well as dendritic cells and neutrophils, and they actually bind to the Fc receptor. The antibody binds to the Fc receptor, and then that triggers the pathogen, or the phagocytosis of the pathogen. So this is showing us IgG. The small monomer antibody has bound the pathogen, and then the Fc stem is bound to the Fc receptor, and that's going to initiate phagocytosis. We've also seen this word opsonization referred to how complement works. So complement proteins can also opsonize pathogens. And remember, I used that analogy where I said it's sort of like candy coating. So if we're candy coating the pathogen in antibody, that's going to attract phagocytes to eat it. All right, fourth function. Those Fc stems, they also can bind to Fc receptors on natural killer cells. So natural killer cells, when they come in contact with Fc stem of antibody, they will degranulate and they'll release their cytotoxins and they'll know to kill that particular pathogen. So that's an overlap into second line of defense, since natural killer cells are second line of defense, but the antibodies are enhancing that second line of defense. Now the fifth thing, and that the fifth function of antibodies is actually complement activation. So complement proteins can be activated if they come in contact with a pathogen. We'll call it antigen. If antigen is bound to antibody, if it's coded in the antibody, then the complement protein comes along and recognizes the Fc stem, and then complement begins to activate. Remember, complement is part of second line of defense, but it helps to enhance inflammation and enhances phagocytosis and also acts as an opsonization molecule as well. So all of that gets enhanced by antibodies being made and produced by the plasma B cells in the humoral response. So that's it about antibodies and their antigen interactions. I hope that was helpful.